Hello everyone, today we're going to talk about eukaryotes. We're going to talk about fungi, protozoa, and helminths. Are basically worms. So let's go ahead and discuss a few vocab terms that we need to know. So we'll start talking about our fungi first. And the study of fungi is known as mycology. Ology means the study of, and myco means fungi. Fungi are actually known to be chemoheterotrophs, which means that they are able to decompose organic matter. Most fungi are actually aerobic, meaning that they do require oxygen. And some fungi out there, they are facultative anaerobes, which means that they can actually survive in conditions that are aerobic or in conditions that are anaerobic, meaning no oxygen, okay? But they actually prefer to live in conditions that do have more oxygen, okay? And actually some of them are anaerobes, so meaning that they don't need any oxygen, okay, for them to survive. Now, fungi, they actually reproduce either sexually or asexually by producing spores. So let me show you right here, okay? So as I was saying, fungi, they reproduce either sexually or asexually by producing spores. And by the way, these spores are not the same as the spores that bacteria would produce, okay? The spores that fungi produce can actually be used for reproduction. But when bacteria produce spores, that's usually when the bacteria are old, or if they are facing harsh conditions, then the bacteria would feel the urge to produce these spores, and those spores act as dormant capsules, kind of like time capsules, okay? And when the conditions are basically okay, then the bacteria spores are going to grow back into the vegetative bacteria, okay? But fungi, their spores are meant, for reproduction. So the spores would actually detach, okay, from the parent cell, from the parent fungi, and they will actually be able to germinate, and you would get a new fungus, okay? Now, if they go ahead and you get these spores reproducing sexually, then what happens is you would basically have two fungi from the same strain, and their nuclei would go ahead and fuse together, okay? So therefore, that's known as sexual reproduction because you have two fungi and their nuclei fused together. That would be the sexual reproduction because it involves two nuclei from two strains of fungi. And again, those strains of fungi could be like two totally different strains of fungi. And that, again, would be considered sexual reproduction. Asexual reproduction, on the other hand, that's when the spores are going to be released and they're just going to germinate into a new fungus, and it's going to be the exact same replica. So right here we have an image of the spore, and you can see like basically what happens when that spore germinates. So right here you can see that this spore right here, when it's released, it basically grows and germinates into a new fungus. So this part that comes out of the spore, it's called the hyphae. And you can see that the hyphae right here basically continues to grow. Hyphae, by the way, it's the fungus, like, structure, and kind of looks something like that. So when you have fungus that's looking like a mold growing on a plate, so if you have a media plate and you have a fungus growing on it and you see that like fuzzy, hairy like structures, okay, what you're basically seeing is this. What you're basically seeing is the hyphae of the fungus. In the hyphae of the fungus, under the microscope, you're not going to be able to see that with your naked eye, but under the microscope, they kind of have divisions like this, and so we call them septate, okay, septate hyphae, or sometimes they don't have any divisions and we call them coencytic, or you could just call them aseptate because A means without. And again, those hyphae, that's where the spores eventually will come out, and I'll show you different images of the spores. And so eventually the spores will come out, and when the spores fall down, those spores can actually go ahead and go through asexual reproduction, as I said, to grow just a new replica of the parent cell. Or, as I was saying, you can have the nuclei of two different strains of fungi, for example, fusing together, and that would be considered asexual reproduction. So when you go ahead, as I said, and take a look at a fungus growing on a plate, on an agar plate, you're going to notice that there is usually that fuzzy, hairy-like structure. It could be white, green, black. Now what you're seeing is a bunch of hyphae, okay, and we refer to it actually as mycelium. So when you see a bunch of hyphae on your plate, for example, like right here, you see that fuzzy, hairy, white structure right there, that's mycelium. Plural, we would call it mycelia, and basically it's just lots of hyphae, okay? Lots of hyphae grouped together. We refer to it as mycelia. And the hyphae that's usually on the plate, like touching the agar, we call it vegetative hyphae. And so that vegetative hyphae is usually accumulated on the plate. And usually when the environmental conditions are suitable, that's when the hyphae is going to start growing, and you're going to start seeing that filamentous mass, right, known as the mycelium. And the hyphae that you would find, as I said, growing on the plate itself, we refer to it as vegetative hyphae. So a bunch of those vegetative hyphae you would see it as the mycelium. And those vegetative hyphae, they're actually the ones absorbing the nutrients from the plate. And sometimes you would also see hyphae that's radiating up, okay? So let me show you right here. So like right here, okay, those we call them aerial hyphae, and they're not really touching the plate, so they're actually sticking up, okay? So that's why we call them aerial hyphae. This is a zoomed in image, by the way, so that you can kind of see the hyphae. They're just very, very thin hair-like structure, okay? And a bunch of them would give you the mycelium, okay? And the aerial hyphae is usually the part that's going to be responsible for the reproduction, because usually the aerial hyphae, that's basically, as I said, projecting above the media surface, this is where you're going to have the spores. And remember how I said the spores is basically for reproduction. So the aerial hyphae is usually the part that's carrying the spores. And again, the spore, like right here, do you see those like black structures right here? This is where you have all the spores. So if this fungus wants to replicate itself, the spores would fall down on the plate and they will start growing into new hyphae, basically into new fungus. Okay, um, so that's, that's how it would look like on the plate. And let's go ahead and take a look at what's next. Those are some terms right here that we usually use when we are talking about fungal infections. So a fungal infection is actually known as mycosis. When you say systemic mycosis, that means it's a fungal infection that's deep within the body of an individual. And it can affect a number of their tissues and organs, so that's a really horrible, infection if it's systemic. Usually individuals, they contract a fungal infection from the soil. For example, if they've inhaled the spores of the fungi, and so if they inhale the spores, they can go ahead and get infected because it's going to move into their lungs. And then basically the infection starts to spread. And those who are more at risk of actually getting affected, when they inhale those spores are definitely those that are immunocompromised. Now, a subcutaneous fungal infection, that's basically an infection that's below the skin surface. And again, usually individuals would contract these subcutaneous mycosis by direct contact, for example, with the spores of the fungus. Then again, it's usually found in the soil. And so usually via direct contact, for example, to a puncture wound that they have in their skin, they would go ahead and get infected this way. And again, can quickly move to the area beneath their skin. Dermatophytes, those are actually usually the fungi that would infect the like the skin, okay, or like the epidermis layer of your skin, the hair and the nails, okay, so we call them dermatophytes, okay, so dermatophyte fungi would be the ones that again would infect the hair and the nails in the skin. Now cutaneous mycosis, that would basically infect the hair and the skin and the nails. So as I was saying, like the dermatophyte fungi, they are the ones that usually cause the cutaneous mycosis. And those dermatophyte fungi, what they usually do is they usually break down actually the keratin protein, which is the protein found in your hair, in your skin, and your nails. So basically breaks it down. And it is pretty contagious, okay? So if you do get in contact with them by touching, for example, infected hair, okay? So if someone has a fungal infection in their hair and you go ahead and you touch it, and especially if you have a puncture wound, for example, then you're going to get infected yourself, okay? Okay, so you can definitely, pick it up from an individual who has it. Now, opportunistic mycosis, those are fungi that are actually harmless as long as they are living in their normal habitat. But then they become pathogenic inside of a host, especially if these hosts are immunocompromised. So your immune system is basically, if someone is immunocompromised, your immune system is not doing that great, and so your immune system is pretty weak. And an example of actually a type of fungal infection that's opportunistic would be yeast infection. For example, Candida albicans. So Candida albicans is a yeast in, like, you know, it's a fungi, it's a yeast. And Candida albicans, again, it's usually not infectious. But then if someone's immune system starts to decrease, let's say it's decreasing because this individual is taking lots of broad spectrum antibiotics, then that can actually start to change their flora, like their gut flora and their flora within the reproductive part of their body, you know, the reproductive organs. And so therefore, this region, especially in females, actually, that would be the vaginal region. The flora within this vaginal region in a female can actually start to change if the female is taking too much antibiotics. And that's when the Candida albicans can actually cause problem, and the female would get a yeast infection. So that's a type of an opportunistic, you know, mycosis. And then what starts happening is, like, you know, that Candida albicans starts to like thrive, you know, within this region, like within the vaginal area, and it causes like symptoms like vaginal itching, etc. It can even cause like discharge. It's usually like white, you know, white secretion. So that's, again, like, you know, an example of an opportunistic fungi. Let's talk about two different types of asexual spores that we have out there. So asexual spores, we are referring to them as asexual because no sexual reproduction is actually taking place. Those asexual spores, they are able to go ahead and grow new fungi, okay, and those fungi would actually be exactly identical to the parent fungus, okay. Now, the two major types of asexual spores that we have are conidiospores and sporangiospores, okay. These ones right here, Arthroconidia, Blastoconidia, Chlamydoconidium, those are all under the group conidiospores, okay? So those three right here are basically subgroups, okay, under that large category known as conidiospores. So let's just kind of see how conidiospores would look like. So this is an example right here. This is a fungus called Aspergillus niger. And when you take a look at Aspergillus niger under the microscope, it would look something like this. You will see these stalks that we call conidiophores. And sticking out of these conidiophores stalks, you would see those chains, kind of like a chain-like, okay, with all of these bead-looking like structures, and those would be the spores. Those are the conidiospores. Now, a bunch of conidiospores, we call them conidia. So conidia is the plural form. Conidiospore would be, you know, one spore, or you can call it conidium. So conidium and conidiospores, the same thing, and a bunch of them would be called conidia. Okay, so this is how, this is one look, okay, for the conidiospore. Now another look, they would look something like this, like little squares, and when they look something like this, like squares, we call them arthroconidia. And Arthroconidia, you usually get this look when the hyphae itself goes ahead and breaks up. So when the septate hyphae kind of fragments off, we end up calling them Arthroconidia. This is another type of conidiospore. It would look something like this. We call it Blastoconidia. Okay, so Blastoconidia. An example of a fungus that actually has this look, and you would see it under the microscope, is Candida albicans, which is actually a type of yeast. Okay. There's also this look right here, Chlamydoconidium, and they look something like this. Very similar to the Blastocanidia look, by the way. And again, Candida albicans can produce this, this type of spore as well, right here. So these again, these are different morphology, okay, for the Canidiospores. Now there's another group called Sporangiospores. And sporangiospores, they are usually encased inside of a sac. Okay, so right here there's a sac called sporangium, and inside of that sac you find your sporangiospores. And this sac right here, it's usually sticking out of a stalk called sporangiophore. An example of a fungus that would form sporangiospores is Rhizopus. Okay, so the genre Rhizopus would go ahead and produce sporangiospores. And those spores, okay, they are all asexual spores. So these spores can go ahead and reproduce and, grow into a new fungus, okay? Of course, the same type of fungus. So it's those spores can go ahead and grow into the same exact identical type of fungus that you have. Now, yeast are non-filamentous unicellular type of fungi. And so when you go ahead and take a look at yeast growth, let's say on an agar plate, you're usually not going to see that mold look, okay, that hairy fuzzy look. It's actually going to look more like the bacteria colonies, okay, on a plate, but they're usually going to be much larger on the agar plate. Now, yeast, they actually go ahead and reproduce by the process called budding. So budding is basically a process where here's the parent cell, and it goes ahead and you start getting that bud coming out of it. And that bud enlarges and enlarges, and eventually that bud goes ahead and detaches off of the parent cell. So now you have a new yeast cell that's basically identical to that parent yeast cell right there, okay? What happens is these buds, if they don't detach though, okay, so sometimes as the yeast produce those buds, they fail to detach themselves from the parent cell. And so you end up with a, kind of like a sort of a short chain of cells. And under the microscope, it's actually going to look similar to a hyphae. Remember those hyphae stalks that we were just looking at? So under the microscope, they would look something like a hyphae, but they're not really hyphae. They're just basically a chain of buds that haven't detached. And so we refer to it as pseudo-hyphae, because pseudo means fake. An example of a yeast that you would actually see those pseudohyphae under the microscope is Candida albicans. So Candida albicans, it's a yeast, and it goes ahead and kind of, produces those pseudohyphae look. And actually, when they produce those pseudohyphae, the Candida albicans is actually going to be able to invade deeper tissues because of, those long pseudohyphae that they would produce, so they would actually be able to invade deeper tissues in your body. Now, some other type of yeast, when they go ahead and they reproduce, we refer to them as fission yeasts because they are able to divide evenly. And so when they come and reproduce, you would get even-sized, okay, you would get even-sized new cells. And so that process is called fission. And basically during that process, the parent cell elongates itself, and then the nucleus divides, and the two offspring cells will be produced. And again, they would be equal in sizes. So that's another, type of reproduction. They go through that fission process. And yeast, by the way, they are capable of facultative anaerobic growth. Okay, so they are capable of growing in conditions high in oxygen or low in oxygen. And so therefore they can survive various environments. Um, and so they can, as I said, grow in high oxygen environments or low oxygen environments, and we take advantage of that because we use yeast, right, to make alcohol, to produce ethanol, because um, um, that process, you would want to put the yeast in a low oxygen environment, and so when they're put in a low oxygen environment, they can go through fermentation, and so that's the process that they use when you're, in brewing and winemaking and even baking industries, okay? And that's why when you add yeast, for example, to your dough, your dough is going to rise, because when yeast are put in a low oxygen environment, they go through fermentation and then they start producing ethanol, and they also start producing carbon dioxide. And that gas, carbon dioxide, is what's going to allow your dough to puff, okay? Now, we have what's known as dimorphic fungi. So there are some fungi out there, and they actually have two forms of growth. So some fungi out there, they can actually grow either as a mold or as a yeast. And so that's why we call them di, because they can, have two different forms. So they can grow in a mold-like, okay, and so they would produce those hyphae on the plate, etc. And they would have that hairy, fuzzy look. And then same fungus can actually grow into a yeast-like form, okay, on a plate. And as I said, the yeast form would kind of look similar to the bacterial colonies, right? You get those kind of, colony looking like type of growth. Now, those dimorphic fungi, okay, they are usually temperature dependent, meaning that usually at 37 degrees Celsius, they would look like yeast, and they would grow in yeast-like forms. Around 25 degrees Celsius, they would look like mold, so you would get that hairy, fuzzy look, okay? Now, actually, humans, our body temperature is 37 degrees Celsius. So a fungus, usually when it grows inside of a human, it would grow in a yeast-like format. Okay, but if you have that same fungus at room temperature growing on a plate, it's actually going to look like a mold-like type of growth. Okay. Um, and so this is basically dimorphic fungi. They can change their growth format based on the temperature. Another group of unicellular eukaryotic microorganisms that we're going to go ahead and discuss are protozoa. Protozoa are normally found in water and soil. Um, Water that could be either fresh water or salty water. They could be located there. Protozoa, if they are found in conditions where the nutrition is low, or they are facing harsh environmental conditions, what protozoa can do is they can go ahead and produce those cysts, which will act as protective capsules. So they go ahead and they produce those protective capsules so that they can go ahead and protect themselves. We're going to talk about the life cycle of protozoa, and usually the life cycle of protozoa, they are pretty complex and it has multiple stages. One of the names that you might go ahead and face when you are reading about a life cycle of a protozoan is the term trophozoid. So trophozoid, that's the stage that we refer to the stage trophozoid. Basically, that's the stage when the protozoa is growing, okay, and feeding. So the feeding and growing stage of the protozoa, we refer to it as trophozoid. Most protozoans are they are aerobic heterotrophs. So meaning that they need oxygen for them to go ahead and grow. Now, why am I saying mostly? because there are some protozoa that are capable of anaerobic growth. And so, for example, when you find someone having, they got infected by a protozoa and that protozoa is inhabiting their intestines, I mean, those intestinal protozoa, they are going to be capable of anaerobic growth. And by the way, there are some protozoa that are actually also photosynthetic. All protozoa, they like to live in areas that has a large supply of water. Um, and protozoa, they do have vacuoles inside of them, and those vacuoles are going to be where the digestion takes place. Okay, so food digestion takes place within those vacuoles inside of the protozoa. And then the protozoa are going to produce waste. So the waste is going to be eliminated either through the plasma membrane of the protozoa or actually through specialized pores, such as the anal pore. Protozoa, they go through the budding process or the fission. So they go ahead and they duplicate themselves. Um, they are basically going to be exact replica of each other. So it's kind of like a cloning process that they go through. Let's take a look at a few examples of some protozoa out there. So again, protozoa, there are such a large, diverse group. So we're just gonna, mention a couple of them. Okay. We'll take a look at group of protozoa that we refer to as ciliates. So ciliates means that these protozoa, they do have cilia. And you can see right here the cilia surrounding that protozoa. And the cilia, those are tiny hair-like structures. And so when the cilia moves back and forth, it can actually help the protozoa move. Okay. An example of a protozoa that does have cilia all around it is Paramecium. Okay, and Paramecium usually find it in lakes and ponds. Um, there is also another example of a protozoa, and this one is amoeba. So this is an image right here of amoeba. And amoeba is a protozoa that doesn't have cilia, but it is motile. It is going to be able to move around, but it doesn't move using cilia. It moves around using what we refer to as pseudopods. Pseudo meaning fake, and pods mean feet. So those pseudopods would help the amoeba crawl, and that's how the amoeba would go ahead and move around. Um, and um, is there are there any pathogenic amoeba, by the way, out there? Yes, there is one called Entamoeba histolytica. And Entamoeba histolytica, it's actually a pathogenic amoeba that can be found inside of human intestines, so would like to reside in a human intestine. And that's one of the pathogenic types that we have. Um, and it does cause symptoms, and you would get sick if you do get infected with the Entamoeba histolytica. Now, there is actually another type of protozoa that we refer to as hemoflagellate. Hemo meaning blood, and flagellate meaning that this type of protozoa would move um, using its flagella. Okay, so that's another type of protozoa, and there is one um, hemoflagellate. And by the way, why is it called hemo, like meaning blood? Because it would reside in the blood. Okay, so how would someone get a hemoflagellate? It's usually transmitted by the bites of a blood-feeding insect. and they would enter into your circulatory system, okay? They would enter into the host's circulatory system, and they use their flagella to go ahead and move around. There's actually a type of a hemoflagellate that causes the African sleeping sickness. So you would always feel very tired, sleepy, and it's usually transmitted. This protozoa is usually transmitted to the human host by the tsetse fly. Okay, so that's really, like, you know, how it's transmitted, okay, by that fly. There's also some that are transmitted by insects that would bite you, or bugs that would go ahead and bite you. So again, you know, that's how mostly humans get infected by these protozoa, through an insect bite, a bug bite, etc. Now the other group that we are going to talk about are helminths. And helminths, those are basically worms. So we're going to talk about two groups of worms out there. We're going to talk about the phylum Platyhelmins, and those are basically the flatworms. We're going to talk about the phylum Nematodes, and nematodes, those are the round worms, okay? So we'll go ahead and take a look at a few examples of those two groups right there. Let's first discuss some characteristics of those helminths. So helminths, those worms, they usually lack a digestive system because they can actually absorb nutrients from their hosts. So they can absorb like the host's body fluids, they can absorb nutrients from the host's tissues. So they can go ahead and really depend, okay, they usually depend on their host. Their nervous system is very minimal because, again, they don't really have to search for food or, you know, respond to a lot of environmental factors. Their movement is occasionally like reduced or actually completely lacking, and that's because they're usually transferred from a host to another, and so again, they don't really need such extensive means of movement. Their reproductive system can differ. So there are worms out there that are, we refer to them as dioecious. So let me write it for you. So if I say this worm is dioecious, meaning that this worm, you actually have a female worm and you have a male worm. The female worm is going to have the female reproductive parts, and then the male worm is going to have the male reproductive parts. Okay, so so again, if I say an adult worm, an adult helminth is dioecious, that means that you have a female and a male. Okay. Now, but if I use the word monoecious, so mono meaning one, and so these worms right here, you would actually find one worm having both parts, okay? So you would have one worm having both the male and the female reproductive parts, okay? You can either call them monoecious, by the way, or you can call them hermaphrodite. So monoecious, or basically, let me write the other word down. So, or you can call them hermaphrodite. And that means that, as I said, they have both the female and male reproductive part in one worm, basically. Okay, so hermaphrodite or monoecious. Okay, and actually the hermaphrodite worms, when they want to go ahead and reproduce, they can basically, you know, they can go ahead and basically like their eggs and their sperm will go ahead and combine, okay? Because I mean they have both, right? So they would just release their eggs and sperm, and it would just fertilize, okay? But if you have you know, a dioecious worm, then, you know, for reproduction to take place, you basically need a male reproductive, you know, a male worm with its reproductive parts, you know, with a female worm that with its female reproductive parts, okay? So let's go ahead and take a look at two phyla, okay, of the helminths. So let's take a look at the worms, the helminths, and we're going to discuss one of the phyla of these helminths, and we refer to this phylum as the Platyhelmins. Now, Platyhelmins, those are flatworms. Now, under the phylum Platyhelmints, there are several classes underneath that phylum. One of the classes that we're going to talk about, it's a class known as Nematodes or flukes. Now, Nematodes or flukes, those have a flat leaf-shaped body. So you can see this picture right here. And they have those oral suckers and ventral suckers. And those suckers are basically going to help hold the organism in place. So it's going to help that fluke kind of attach, okay, to surfaces or basically attach to its host so that they can stay in place. Now, how would a fluke absorb its food? Flukes usually obtain their food by absorbing it through their outer covering. Okay, so this outer covering right here, we refer to it as the cuticle. Okay, and they can go ahead and absorb the food through their cuticle. Flukes, by the way, are usually given their name, their common name, okay, not their scientific name. Their common name is usually given according to the tissue of their host, okay, or the host where they're going to grow into adults, by the way, okay, so this is how we give them the common name. So for example, if I say lung fluke, the reason why I call it lung fluke, and that's the scientific name, by the way, it's the genre right here, that's the genus name. This one, though, the common name, let's concentrate first on the common names. This common name is called lung because it's going to reside in the lung tissue of its host, and this is where it's going to go ahead and you know, grow into an adult worm. There are liver flukes, and those usually reside in the liver of the host. There are blood flukes, and they reside in the circulatory system. Okay. Now let's take a look at this, you know, this genus right here, and it's called Paragonemus. Now, Paragonemus, that's the genus, and there are multiple species, okay, of the Paragonemus fluke. Now, one of the Paragonemus flukes out there, it is located in the United States, so there are cases of people getting infected with the Paragonemus, you know, Tremadode in the United States, and it's usually, how do people get it? It's usually associated when you eat raw crayfish, okay? So usually when you eat raw crayfish, and the crayfish does have the eggs and the larvae of those flukes, and you go ahead and you ingest it, and then it basically grows inside of you, and then it goes all the way to your lungs, okay, and it would reside in the lungs, okay, so it would actually reside in the lungs and in the bronchioles, the branches of your lungs, and it's approximately about 12 milliliter long, okay, so each fluke would be 12 milliliters long, but of course, once it resides in your lungs, I mean, you're not just going to have one of them, right? They can actually go ahead and reproduce into, you know, multiples, okay? So usually, how would we figure out whether someone has been infected with with this lung fluke? We take a sample of their sputum. Okay, so you take a sample of their sputum, and if you actually find eggs in their sputum, that's an indication. Okay, so you would actually secrete eggs. There would actually be eggs in their sputum. And by the way, those eggs also, so if you do have this parasite inside of you, you can also be excreting the eggs in the feces. Okay, so usually the eggs are going to be excreted in the feces of the host. Okay, and those eggs, okay, that you secrete in the feces, let's say due to eating or drinking water or food that are contaminated with fecal matter, this is again how you can get infected, okay? Or if you eat, as I said, like raw seafood, especially raw crayfish, okay? Um, and if you take a look actually at this lung fluke, it is monoecious. So it doesn't need a partner, right? It has both the male and the female reproductive parts. And so especially when you have a monoecious, parasite inside of you, um, they're going to reproduce pretty fast because as you can see, they have both the parts. So they're going to multiply really fast inside of your body, okay? And as I said, to diagnose it, we can take a sample of the patient's sputum or feces, okay? And what we're trying to find or locate, we are trying to find the fluke's eggs, okay? So this right here kind of shows you the complex actually life cycle of the lung fluke. And so basically the infection all starts from eating, as I said, undercooked, for example, freshwater crustaceans, especially crayfish, and or if you eat like completely raw or basically crayfish or freshwater crabs that are not cooked properly, what ends up happening is you go ahead and as you eat that raw crayfish, you're basically going to swallow, okay? You're going to swallow the lung fluke, but you're not going to swallow the adult stage of it, okay, guys? So you kind of swallow like this stage of it right here. But what ends up happening inside of you is those hermaphroditic, adult flukes are going to start releasing the eggs into your lungs. They're going to start, growing inside of your lungs. And eventually, as I said, you can release those eggs in your sputum or in your feces, okay? Now, if a patient, goes ahead and releases the eggs in their feces, and those feces make their way into water, then those eggs are going to get into a snail, okay? Now, these snails are what we refer to as intermediate hosts. There's another intermediate host, and that would be the crayfish. What are intermediate hosts? Intermediate hosts are basically hosts for the lung fluke, and this is where the lung fluke is going to go through asexual reproduction. So intermediate hosts, that's where asexual reproduction takes place. We humans are actually the definitive host for the lung fluke, meaning that we humans are going to be their site where they're going to go through sexual reproduction inside of us. Okay, that's the biggest difference between a definitive host and an intermediate host. Intermediate host, this is where asexual reproduction is going to take place. Definitive host, that's where sexual reproduction is going to take place. Now, are we humans always definitive hosts for parasites? No. Okay, sometimes we actually act as an intermediate host. Um, so is it important, by the way, before they reach humans, for them to actually go through their life cycle by entering into an intermediate host first? Yes. Okay, it is pretty important for them to actually follow their life cycle in order. So they will have to find, those intermediate hosts before they can go ahead and actually like infect us. Otherwise, you're kind of breaking the cycle in the middle, and it's not going to work. It's not going to go through. It's not going to complete. Okay, it's not going to be completed. So one way actually for us to get rid sometimes of pretty nasty parasites that are out there is take out their intermediate hosts. Okay, so take out their intermediate host, and what you're going to do is you're going to go ahead and make the cycle incomplete, so they're not going to harm us. Okay. So this is one example, okay, of the lung flukes. Let's take a look at another example. So let's go ahead and actually take a look at another group or another class under the Platyhelmins, and those are called cystodes, or we refer to them as tapeworms. And those are intestinal parasites. So you would find them in, within the intestines. This is where they like to reside. They have heads, okay, and this is like a enlarged image of their head, and we call their head scolex. And they have suckers right here, okay, and those suckers are going to actually help them attach to our intestinal mucosa of their host, okay. And sometimes they have small hooks. So sometimes they even like have those small hooks right there, and they basically pierce those hooks, okay, in the mucosal lining of their host. And again, why are they doing all of this? Because they want to attach. They don't want to let go. Tapeworms or cystodes, they don't ingest the tissue of their host though, okay? They don't have a digestive system. So to obtain nutrients from the small intestine, they would actually absorb it again. They would absorb it through their cuticle, okay, through their outer covering. When you take a look at their body, see this is their body right here. Notice how their body is basically segmented into these like squares, okay? We call those segments proglottids. Okay, so proglottids, that term that you're seeing right here, that's basically those segments, okay, that you see within their body. Basically, these proglottids, okay, each mature proglottid will actually contain, okay, so if we zoom in on the proglottid, what would you find in there? You would actually find both the male and the female reproductive organs. So again, this is an example of a monoecious, right, worm. So this is a monoecious tapeworm, and again, it has both the males and the female reproductive parts. And so again, if this tapeworm gets inside of a host, it can go ahead and multiply pretty fast because it has both reproductive parts. And so what ends up happening is, as fertilization takes place inside of each proglottid, each one of those, right, each one of these proglottids are actually going to contain lots and lots of eggs, okay, which is pretty bad for the host, okay, because these eggs will, these proglottids will kind of burst, and then those eggs are going to be released inside of the host, and it's going to grow into more and more worms, okay. There are multiple types of, multiple examples of tapeworms out there. I mean, there's the beef tapeworm, and the beef tapeworm, again, it's called beef because usually when you eat, again, raw meat, basically, okay, especially if that raw meat has the eggs, okay, of these worms, and you go ahead and you eat it, you will get infected. And beef tapeworms can reach 6 meters long, okay, inside of your body. So there are multiple examples, but this is kind of like, one example out there. And usually, how do we diagnose someone, okay, if they have, let's say, a tapeworm infection? So basically, we try to find those eggs in their feces. So you would take a sample of the patient's, fecal matter, and you go ahead and you see if they have those eggs or not. And that's why, by the way, like, beef has to be inspected very properly and, make sure that it's not, doesn't carry any of the, tapeworm eggs, etc. And that's why, again, it's recommended, okay, not to eat raw, raw meat. There is one, so you're like, okay, well, I'm gonna steer away from beef and I'm just gonna eat pork. Well, same problem. So there is also pork tapeworm. So also, again, do not eat raw pork because, again, you could ingest those eggs, and once you ingest those eggs, it is going to start growing inside of you. Um, and again, that's why it's important not to eat undercooked meat. That's why it's important not to eat, contaminated food. Okay, because again, if the food is contaminated with someone's fecal matter, again, if someone is infected and they use, let's say, their restroom and they don't wash their hands, well, guess what? They could have eggs underneath their nails, and then if they cook the food, etc., they can go ahead and deposit those eggs in the food, and then you end up eating it. So again, this is how commonly humans get infected. Okay, they ingest these eggs right there. So that was just, a couple of examples for you. Now, we also have another group, and those are the nematodes. So nematodes are another phylum, okay? So far we talked about the Platyhelminths phylum, and the second phylum that we're going to talk about are nematodes. Nematodes are roundworms. So they're kind of cylindrical, okay, and they do have a digestive system. They do have a complete digestive system. They do have, like, a mouth and an intestine and anus and everything. And most of them are dioecious, so they're males and females, okay. So these are a few examples right here of some nematodes that can actually infect us humans. And so there is the pinworm. There is, like, the ones that would inhabit your intestines, the Ascaris worms. So the Ascaris worm actually, Ascaris usually infects around 1 billion people worldwide, okay, and it is dioecious. It has a female and a male part to it. And it would usually, like, if it gets inside of a human, it would actually reside in the small intestine of humans. And this is kind of like their exclusive place. They like to reach your small intestines, and they like to feed on, like, the semi-digested food that you have in your small intestines. And again, as they're growing inside of your intestines and they start releasing their eggs, usually, as the human host defecates, the feces will be carrying the eggs, okay? And by the way, like Ascaris, they can survive in the soil for a pretty long time. So that's, that's another location you would find them, okay? If they're not in the small intestine of a host, they might be found in soil. They can, as I said, like, if you eat, again, contaminated food that's contaminated with fecal matter, etc., you go ahead and you get exposed to them, and then you swallow them, and then you go ahead and get infected. Now, even sometimes, by the way, like when people cough, okay, they can cough out larvae, okay, of those worms. So not something, like, that would be good at all, okay, for you, because, I mean, it will just, like, take over your lungs, and it's going to be extremely hard for someone to breathe. It's going to feel like there is extreme, like, something compressing their lungs, basically, okay. It's gonna, it's not gonna, unfortunately, though, you're like, okay, well, can someone detect it pretty fast? Unfortunately, that's not the case. So by the time you detect that you are infected, they've already, taken control of the lungs, and they're already spread. Okay, so to get rid of them, it's going to be a pretty hard task. Okay, there is another one that we can go ahead and talk about, and it's that pinworm right here. And so pinworms, those actually spend their entire life in the human host. Okay, so they want to find a human host, and then once they find a human host, they're pretty happy and they can actually stay their entire life there. Those adult pinworms, you would find them in the large intestine of the human host, and what happens is those pinworms, by the way, are dioecious. So there's a female and there's the male, and so the female pinworm, what it does is it can actually migrate, usually migrates to the anus. Okay, so migrates to the anus. And after it migrates to the anus, it goes ahead and it deposits her eggs on the skin of your anus. So they're inside of you, okay? So they're inside of your intestine, okay? The worm makes its way through your intestine, okay, guys? It kind of reaches the rectum right here, and it reaches the anal, okay? And then as it reaches that anus part, it goes ahead and it deposits its eggs, and it will actually deposit it outside. So the eggs are going to be like right here on the butt cheeks, okay? And it deposits it there. What happens again? This is how another person would get infected with it. Is when the female pinworm releases the eggs, and now they are deposited on the skin, this area is going to get really itchy. Okay, so if it gets really itchy and the person is going to start scratching this area right here, what ends up happening is the eggs get underneath the fingernails. Okay, and so the thing is, you can reinfect yourself. How can you reinfect yourself? Well, if you scratch this area, and let's say it's hygiene is not too good, and you don't wash your hands, and then you have now eggs underneath your fingernails, and then you put your fingers in your mouth, well, you've just re-swallowed some eggs. Okay, so the problem keeps going. And then again, if you scratch this region right here, and then you touch food or cook or whatever, you can go ahead and infect others this way. So kind of a good thing is always make sure that you wash underneath your fingernails as well, okay, because there could be tiny eggs underneath your fingernails and you don't know, and you go ahead and you get infected. And so again, that's why also the bed sheets, okay, gotta be careful with bed sheets, because if someone has been if someone has used those bed sheets before you, and again they are infected, well, those eggs are going to be all over the bed sheet, and you're not going to see them, by the way, with your naked eye. Okay, so they're too small, and you're not going to see them in the naked eye. And by the way, a lot of kids too get infected with pinworms, because those eggs can be also in the sand, and so when kids go and play in the playground, etc., kids tend to put their hands in their mouth a lot. They can actually ingest those pinworms. And, so actually, the doctors would tell you that if you are suspecting that your kid might have pinworms, because this their buttocks area is always itching them, what you could do is actually get tape, okay, and put the tape on their buttocks area, and at night, usually that's when the female pinworm likes to go and deposit its eggs. So you can go ahead and check the tape in the morning, and if there are eggs attached to the tape, then this is confirmation that your kid has pinworm infection.